Good morning, all. Today, we are going to discuss about immunity, structure, and function of antibodies. First of all, we will discuss about immunity. So, what is immunity? Immunity is the resistance offered by the host against microorganisms or any foreign substances. There are two types of immunity. First one is innate immunity, that is the body's first line of defense against invasion of microorganisms. They may be either through virtue of genetic makeup or right from the birth. Second type of immunity is acquired or adaptive immunity. This is acquired during the course of life. It may be either due to an infection. Next, we will look out the types of innate immunity. Innate immunity are of three types: species immunity, racial immunity, individual immunity. What is species immunity? It is the innate immunity towards a microbe exhibited by all members of given species. Example, frogs are resistant to Bacillus anthracis while toads are susceptible. The mechanism may be due to physiological and biochemical differences between the tissue of host species. Next type of innate immunity is racial immunity, that is the innate immunity confined to a particular race. Example, Negroes of America are susceptible to TB than whites. Next, the third type of innate immunity is individual immunity, that is different innate immunity is seen in different individuals. Example, homozygous twins exhibit similar degree of resistance or susceptibility to lepromatous leprosy and TB than heterozygous twins. So innate immunity, two types. So immunity is of two types, innate immunity and acquired immunity. Innate immunity is of three types, that is species immunity, racial immunity and individual immunity. Next we will move on to the mechanism of innate immunity. So the mechanism of innate immunity is by receptor interaction, that is attachment, the binding of surface molecules of microorganisms and receptors of immune cells, that is the primary step, that is attachment. Microbes associated molecular patterns, these are repeating patterns of conserved microbial surfaces, for example peptidoglycan, lipopolysaccharides, teichoic acids, lipoproteins. And there are pattern recognition receptors. These are conserved molecules present on the surface of host phagocytic cells. Example, toll-like receptors. So there may be a receptor interaction. That is, this pattern recognition receptors will interact with the microbe-associated molecular patterns. That is the primary step. The second step of mechanism of innate immunity is activation of transcription factors. That is, this binding will lead to the generation of signals for the activation of transcription factors for expression of genes encoding cytokines and enzymes, thereby possessing antimicrobial activity. For example, NF-kappa B, it promotes the production of cytokines, interferon regulatory factors that will promotes the expression of antiviral interferons alpha and beta. So up to this, we had discussed what is immunity, what are the two types of immunity, that is innate immunity and acquired, what are the three types of innate immunity, that is species immunity, racial and individual immunity, and later on we had discussed the mechanism of innate immunity. Next, we will move on to the components of innate immunity. The components of innate immunity are anatomical and physiological barrier, phagocytes, natural killer cells, mast cells, dendritic cells, complement pathways, inflammatory response, cytokines, acute phase proteins, and normal resident flora that we will discuss later. Next is components of acquired immunity. Components of acquired immunity involve T cells and B cells, classical complement pathway, antigen presenting cells, cytokines which include interleukin 1, 2, 4 and 5. So next we will move on in detail what is innate immunity. Innate immunity may take place either non-specifically or through specifically. Non-specifically, it is the external defense mechanism, that is the first line of defense, and internal defense mechanism, that is phagocytic cells and antimicrobial substances in blood. And specifically, the innate immunity is through the activation of tissue factors, that is the second line of defense, cellular and humoral responses. So we will discuss the first line of defense, that is a physical barrier, which include skin and mucosal surfaces, secretory products of mucosa, and physiological barrier. So intact skin barrier, it mechanically prevents the entry of microorganisms by producing sebum containing antimicrobial peptides, fatty acids, and propionic acid by commensal bacteria. Next, we will move on to the mucosal barrier, that is a mucous membrane, which prevents the entry of microbes mechanically, and mucus entraps the microbes. Cilia in the lower respiratory tract propel the mucus outside. Cuff reflex is an important mechanism in innate immunity. The normal flora of intestinal and respiratory mucosa, these are lined by normal flora that also provide some sort of innate immunity. Next is secretory products of mucosa, which include saliva, tears, and lactoferrin. Then gastric juice. So gastric juice that will help to provide innate immunity, that is low pH, and enzymes of stomach keep the number of microorganisms low. The enzymes include trypsin, bile salts, then fatty acids are there, then gastrourinary tract. That is the normal flora of urine flushes the urinary system. Physiological barrier include temperature, low pH. Temperature, the normal body temperature inhibits the growth of some microbes, and the low pH mainly in the gastric juice, that is gastric acidity, and low pH of vagina due to fermentation of glycogen in epithelial cells prevents microbial invasion. Next, we will move on to antimicrobial substance and phagocytic cells. Antimicrobial substance include complement system, which causes bactericidal activity. Interferon, these are proteins against viral infection. Properdin, that is complement-like substance present in normal serum. Phagocytin, that is heat-stable bactericidal protein derived from polymorphs. Polypeptides like plakins present in platelets, leukins present in leukocytes, they all possess microbicidal activity. Beta-lysine shows activity against Bacillus anthracis. Hydrolytic enzymes and bile in small intestine provide protection from various microorganisms. Then bactericidin is a non-specific serum factor active against Neisseria and Streptococci. Now we will move on to phagocytic cells, which include neutrophils and macrophages. Phagocytic cells reach the site of inflammation attracted by chemotactic substances and ingest particulate materials. So we all know what are the steps in phagocytosis. The natural killer cells, they are a class of lymphocytes that kill virus-infected cells and tumor cells. Intraepithelial lymphocytes present in the epithelial lining of skin and mucosa. Natural killer T cells present in lymphoid organs and epithelium. Then mast cells present in the lining of respiratory and other mucosa. Dendritic cells, these produce numerous cytokines to initiate inflammation and transport of antigens from skin and mucosal site to lymph nodes. That is antigen presentation. Next comes the cytokines that are produced by dendritic cells, macrophages, and other cells in response to microbial antigens, which mediate cellular reactions of innate immunity. Complement pathways, mainly the alternate pathway and mannose binding pathway, they promote innate immunity. Next, inflammatory responses. Biological response of vascular tissue to microorganisms or foreign antigens, which include vasodilation, leakage of plasma proteins through blood vessels, recruitment of phagocytes, engulfment by phagocytes, and destruction. Acute phase proteins. These are proteins synthesized primarily by liver cells because of infection or tissue injury, increased or decreased during acute inflammation, having antimicrobial and anti-inflammatory activity. Main important example is C-reactive protein. So this is all about innate immunity. So we had discussed types of innate immunity, mechanism of innate immunity. Later on, we had discussed about in detail the innate immunity. Next, we will move on to the second type of immunity, that is acquired or adaptive immunity. This is the resistance against the infecting foreign substance that an individual occurs or adapts during the course of life. That is very important. Two types of acquired immunity, active and passive. Active immunity, that is induced after contact with foreign antigen. It involves active participation of host immune response, leads to synthesis of antibodies and production of immunologically active T cells and B cells. This active immunity is a long-lasting immunity and shows two types of responses, primary immune response and secondary. Primary immune response, it is the immune response occurring for the first time, and secondary immune response, this is the immune response occurring during exposure to microbes. So this active immunity is of two types, that is natural immunity or artificial. That is natural active immunity. It occurs following clinical or subclinical infection, example measles viral infection. Then artificial active immunity, it occurs following a vaccination. So next we will discuss about passive immunity. So acquired immunity is of two types, active immunity and passive immunity. So passive immunity, it's a ready-made immunity passively transferred to a host by transfer of antibodies or immune serum. They have no active role of immune system with immediate protective mechanism and short-lasting protection. Useful when instant immunity is required. This is of two types, natural and artificial. Natural passive immunity is either through placenta or breast milk. Example, transfer of maternal IgG or immunoglobulin G to fetus transplacentally, or transfer of maternal IgA or immunoglobulin A to infant through milk. Artificial passive immunity is through immune serum or immune cells, that is ready-made transfer of commercially prepared immunoglobulin. Example is rabies immunoglobulin, human immunoglobulin against tetanus, hepatitis B, diphtheria, botulism, rabies and measles. So this is all about introduction to immunity. So we had discussed what is immunity, then what are the two types of immunity, that is innate immunity and acquired immunity. So in innate immunity, there are three types, species, racial, individual. Later on, we had discussed the mechanism and innate immunity in detail. Acquired immunity is of two types, active and passive. Active immunity is of two types, naturally active immunity, then artificially active immunity. Passive immunity. Passive immunity is of two types, naturally passive immunity and artificially passive immunity. This is all about immunity and its type. It's very important in immunology. Now we are going to discuss about antibodies or immunoglobulins. We all know antibodies or immunoglobulins are specialized glycoproteins produced from activated B cell, that is plasma cell, in response to an antigen. These antibodies will react specifically with the antigens which stimulated their production. Antibodies constitute 20 to 25% of total serum proteins, and there are 5 classes of antibodies: IgG, IgM, IgA, IgD and IgE. Next we will discuss about the structure of antibodies in detail. So coming on to the structure of antibodies, they are Y-shaped heterodimer composed of 4 polypeptide chains. They have 2 identical light chains with molecular weight 25,000 daltons, 2 identical heavy chains with molecular weight 50,000 daltons. These chains are bound each other by disulfide bond and non-covalent interactions like hydrophobic bonds, salt linkages and hydrogen bonds. All the chains, that is heavy chain and light chain, have 2 ends, amino terminal end and carboxy terminal end. There are 5 classes of heavy chains: alpha, gamma, mu, delta and epsilon, and 2 classes of light chain, that is kappa and lambda. Mainly, these antibodies or immunoglobulins are of 5 classes based on amino acid sequence of heavy chain. They have a variable and constant region. Each heavy and light chain comprises of two regions. The first one is variable region. That is the first 110 amino acids near the amino terminal end of both light and heavy chain constitutes the variable region, and we can denote them as VL and VH. V stands for variable, L for light, V stands for variable, H for heavy. This variable region represents the antigen binding site of the antibody. There are hypervariable regions, that are zones within the variable region that shows relatively higher variability in amino acid sequence. They form the antigen binding site. So this hypervariable regions form the antigen binding site, and there are 3 hotspots on L region, that is light chain, and 4 hotspots on heavy chain. Next is paratop. What is paratop? It is a site on hypervariable region that make actual contact with the epitope of antigen. So this paratop of antibody will make contact with the epitope of antigen, and antigen-antibody reaction takes place. The second region is constant region. That is the remaining part of the molecule other than variable region. Here the amino acid sequence is in uniform pattern. Then next we will discuss about heavy and light chain domains. Heavy and light chain regions further folded into domains containing 110 amino acids. Within the domain, a loop-like structure with 60 amino acids formed via interchain disulfide bond. So in light chain, there is one variable domain and one constant domain, and in heavy chain, there is one variable domain and 3 to 4 constant domain. Next, we will discuss about hinge region. So in the heavy chain junction formed between the CH1 and CH2 domain, that is constant heavy chain 1 and constant heavy chain 2 domain, where proline and cysteine is rich, and that region is called hinge region. It's a flexible region. It is sensitive to various enzymatic digestion. So when an antibody is subjected to enzymatic digestion, it will generate various fragments. Example is papain digestion. So if an antibody cleaves at a point above the disulfide bridge of hinge region, it produces 2 FAB region and 1 MC region. That is, FAB means antigen binding fragment and FC means crystallization fragment. So this is the structure. It's a Y-shaped region with a variable chain and a constant region. Heavy chain is there, light chain is there, antigen binding site, that is FAB. Hinge region, it is shown in the diagram, disulfide bonds and all. This is the structure, typical structure of antibody. Next, we will discuss about the functions of immunoglobulins. So what are the functions of immunoglobulins? First function is antigen binding. So antigen binding function is by the FAB region. Binding of antigen, that is a primary function, result in the protection of host. FAB fragment bears variable region involved in the interaction with antigen that we had discussed earlier. Valency of antibody refers to number of FAB region, and for a simple monomeric antibody, the valency is 2. Second function is effector function by FC region, and it includes fixation of complement, binding of various cell types. Antibody coating the target cell will bind to the complement through its Fc receptor, and it will lead to complement-mediated lysis of target cell. And binding to various cell types, phagocytes, lymphocytes, platelets, mast cells, eosinophils, and basophils bears Fc receptor that will bind with the Fc region of immunoglobulins. This binding can activate cells to perform biological functions. So this is a figure showing the function of antibodies. Either it can activate complement, fixation of complement, or binding to various cell types. And second function is antigen binding. So this is all about the structure and function of antibodies. So first of all, we had discussed antibodies. What are antibodies? Types of antibodies, then structure of antibodies, and function of antibodies. Hope you all understood the topic: immunity, structure and type, structure, types and function of antibodies. Thank you.